Okay, yesterday we left off and we had been talking about adaptive radiations. The idea that you can have one species, one common ancestor, and it can spread out over time, especially in the example of mass extinctions. If you have a mass extinction event, then there's going to be areas of the environment, of the world, really, that are suddenly accessible where before they weren't because of the fact that there were predators there were other people occupying that space, and there just wasn't enough territory. So now you have a situation where there's a mass explosion of species over time because there's all of these dominant species that are no longer around. Now I want to shift gears a little bit and talk about another thing that can affect selection, and that has to do with body plan genes. And we talked a little bit about this before, the idea that there are, during development, these genes called homeotic genes, which basically tell an embryo, here's where my wings are supposed to be, here's where my legs or my limbs are supposed to be, here's where my flower petals are supposed to be, etc. In animals, we call those genes Hox genes. We don't call them homeotic genes, we call them Hox genes. And if these are made in the wrong location, or if they're not made at all, that can make a big difference in the survivability of the organism. And it could be in a good way, or it could be in a bad way. So this is a picture showing antennae that were the genes for antennae were moved into the locations where the genes would the cells would normally produce antennae. Instead, they are producing leg segments because they had turned on these genes for these leg segments in these cells in the eye. And this was just done to show that this is a possibility, not just for, they didn't do it just for kicks. We know that during a lot of the mass extinctions afterwards, there was development of critters. During the Cambrian explosion that followed after the last mass extinction, there was development of the mammals and other animals as well. And we know that there were, or at least we think that the Hox genes were pretty important in that. And that could be because of a change in gene sequences, but it could also be because certain genes get turned on or turned off. So, for example, I've got this little guy, this crustacean, that has all sorts of limbs, probably not the most efficient, not the most necessary. And they actually, in lab, did find that you could turn off some of these genes and get the production of segments, or limbs, I should say, limbs that were more like the current day, just having 6 pairs of limbs rather than, let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Lots and lots of pairs of, they just call it many-legged crustaceans. So that could change the body plan, and if you get that dramatic change in body plan, that might be, it might be a disadvantage, but it could also be an advantageous adaptation. All right. Last note for chapter 25 is the idea that evolution is not goal-oriented. It's not trying to make a better thing. It will happen that way, because if it's not a better makeup or a better better adaptation, then it's probably not going to be as successful. It won't survive as much, and therefore it's less likely to pass on its traits. So lots of different things are tried in terms of evolution, and then what is the most, what is the best at reproducing is going to be the one that lives and thrives and passes on its genes in any one particular situation. Okay, so our next chapter is going to be chapter 26 on phylogeny, and let's talk a little bit about what that means. Phylogeny is a way of trying to explain how different species are related to each other, or how they evolved over time. So we can figure out, how did legless lizards and snakes, how did they evolve from a common lineage of legged lizards, for example. And here we've got, this is called a cladogram. or a phylogenetic tree, and we're going to be spending some time on those. The phylogenetic tree shows that there was a common ancestor here. And let me just get a pen here. So I've got a ancestor, and then at some point, branching off from all of these guys were our snakes. And so we have the snakes with no limbs, we have the geckos that are limbed, and all the other ones as well here. And then we have another point where a different set of species called the glass lizards developed, and they also had no limbs as all. So that as well. So that's an example of that. We'll come back to this in just a second, because I went through that. That's just a kind of a teaser, so we'll come back to that in a second. But first of all, let's talk a little bit about binomial nomenclature, and that's the idea. This was one of the first systems that was used to try and classify species and try and see how they were related to each other. So when we talk about classifying things, we talk about a genus name and we talk about a species name, and this is where, if I talk about Homo sapiens, I'm going to capitalize my H for homo, and then I'm going to have a lowercase that's supposed to be an S, sapiens. Okay, so, and then the whole thing would be underlined, or now that we have computers, we can just use italics. Okay, but that tells me that's a genus and species name, and that's a specific organism. And that was used for many, many years, but we realized that that wasn't always the best way to do things. So, but let me run through this. I forgot I got a little bit more before I get to that point. Okay, so we already talked about genus and species, but this this way of classifying things, this taxonomic group system, it starts with a very broad category that contains many, many organisms, and comes down to kingdom, phylum, class, order, family, genus, and species. This is only a way to classify how species are currently similar and different from each other. It doesn't show any evolutionary connections, and so that's why this is sometimes not considered the best way to do things, because we don't want to know how things are connected now. We want to know how things were, how they came to be the way they are, and how they are connected to other species in terms of their evolution. Here is an example of starting off at the domain and working your way down to species, using that taxonomic system. Notice how there are three domains here. There's a bacteria, an archaea, and a eukarya. And I think we talked about these before, but we'll just go over them really quickly now. Our archaea and our bacteria, these are both prokaryotic kingdoms, sorry, not kingdoms, domains. And bacteria has our common nasty bacteria that we think of that causes infections. And archaea also has some prokaryotes in it, but these guys are typically not disease-causing. They tend to be what are called extremophiles, meaning that they live in high salt concentrations, or really, really high heat, or really high pressure. So they have a slightly different domain for that. And then we have domain eukarya. And of course, those are our eukaryotes, and there's only one eukaryotic domain. Then when you look below that, going a little bit more narrow, and you can think of this, they don't do a great job of showing this here, but it's going to be really broad at the bottom, and it narrows down to a species. Below domain, you have kingdom. In this case, it would be kingdom Animalia if we're talking about a panther. Okay, so kingdoms are plants, animals, protists, fungi, and what am I missing? Plants, one more time. Plants, animals, fungi, protista, and oh, well, the other kingdoms are prokaryotic kingdoms. Sorry, forgot about that one. Okay, so that is a way of showing how similar species are, but it's not a way of showing anything in terms of their evolution. Okay, and what we want to talk about, and we want to spend the rest of the time talking about, is the evolutionary history of a group of organisms. All right, sorry about that little interruption there. So we want to focus on what are called phylogenetic trees, and phylogenetic trees are going to get at, or attempt to get at anyway, the evolutionary relationship between groups of animals. Here's another phylogenetic tree between all of these have a common ancestor here, but they all are different, obviously, from one another. Okay, so let's stop talking about classification and talk about phylogeny instead. So phylogeny is, first of all, we need to be very aware that these are hypotheses about the evolutionary relationships, because we can only look at how the organisms are currently related to each other and what they look like today. We can look at fossils, we can look at DNA sequences, we can do other things to kind of give an idea of how they might be evolutionarily related, but they are guesses. They're good guesses, but they are nevertheless guesses. What we can see is that these are showing us where these organisms diverge from each other. So they're showing a common ancestor, and they're showing a branch point, and that branch point is a particular trait, and that particular trait is going to be one where one group has it and one group does not. They both came from a common ancestor, but one group has the trait and one group doesn't, and that's the divergence of two particular groups away from each other. So here I have an example of that, where I've got a common ancestor, and there's my common ancestor here, and then I have fish, I have frogs, and I have lizards and chimps and humans. They all diverge at some point from each other. Notice how the distance is the same in each of these cases. And that is because these phylogenetic trees don't say when this happened. There's not a timeline here. It's just saying that they did diverge and go into their separate groups, and they are related in some way, but it's not saying how long ago that happened. So that's an important point to keep in mind when we look at these things. Here's another couple of them, and sometimes they're done in a diagonal fashion, sometimes they're done in vertical. We can see that chimps have a closer common ancestor than, say, lizards with chimps or frogs with lizards do. So that's one way of looking at it. We can also look at it in a diagonal fashion. We can also rotate around a branch, so I can flip the way these are shown. I could have lizards kind of in the middle here, or I could have lizards flipped, and with the chimps and the humans kind of in the middle there, and the lizards down here. It's still showing the same thing. It's just a different. It's almost like saying I'm gonna talk about something and use different words, but I'm still showing exactly the same relationship. I'm just showing it in a slightly different way. Okay, so phylogenetic trees show patterns of descent, but they do not say when. That's important. They do not say when the evolution happened or how much change has occurred in the lineage. So just keep that in mind. All right, I'm going to skip down here a few slides to get to the first 26.2 section, because now I want to talk a little bit about how we can get a phylogenetic tree. What information do we use to try and figure out how these different groups are related to each other? And there's a whole bunch of things that we can use. We can look at their DNA. We can look at the genes that and what they produce, what proteins they produce. We can look at structure, as well as we can look at the biochemistry, and that biochemistry would be like what do those genes, what types of proteins do those produce, and then therefore what do those proteins do. So that's the biochemistry part. Let's talk about these. Okay, so one thing we can look at is homologies, and this could be either in structure homology or it could be in molecular homology. So organisms with similar morphologies or DNA sequences are more likely to be closely related. That kind of makes sense, right? If you have a similar appearance, in general, it's not always the case. But if you tend to look the same, or if you have very similar DNA sequences, then you're going to that those two groups of species are probably more closely related than ones that have fewer similarities. Those are called homologies. One of the things that we have to worry about when we're doing this, though, is the difference between what's called a homology, which we talked about, that's similarities. That would be like we talked about the evolution and evidence for evolution, and how one bone, two bone, many bone, and how that is evidence for evolution. That is homologous, meaning that a bird wing, and a whale flipper are homologous to each other. They have the same structure, but they do a different function. That tells you they must have come from a common ancestor. Different from that is something that's analogous. And so when you're talking about analogous, you're talking about homologous structures, sometimes they're not homologous. Sometimes they look the same, but they actually, and they have the same function, but if you look at them carefully, their structure is actually very different. So the example for an analogy would be where you have a bat wing, say, and you have an insect wing. They both allow the organism to fly, but they don't really have much in terms of the way of a common ancestor. Okay, so if there's no common ancestor, it's going to be called an analogy. And the reason those develop is because of what's called convergent evolution, meaning that we talked about there's always, in an environment, there's kind of a best way to deal with the environment. So you could be in a completely different environment, and it's still, not completely different environment, a completely different place in the world. But if the environment is the same, it may cause the population to be pressured into evolving in a certain way. So you get, you get flying as a way to really be successful, even though there's not a common ancestor between those species. And so convergent evolution occurs when the similar environmental pressures and natural selections produce something that's similar, adaptation-wise, but it came from different evolutionary lineages. To illustrate that point, if you look at this next slide, I've got a couple of what are called, quote, moles. I don't know why the one is in parentheses. They look like they're identical to each other, but one came from the continent of Africa and one came from the continent of Australia. So very different species in terms of their geographic location. And as it turns out, they're really not, when you start looking at their DNA, they're not very closely related. So that's an example of analogous structures, and just the fact that their environment means that they tend to turn out the same, even though they're in very different locations in the world. Homology can be distinguished from analogy by comparing fossil evidence and degree of complexity. The more elements that two structures have in common, the more likely that they are homologous. You can also look at the genes as well. Genes is probably a better way of doing it, but sometimes a little more complicated to do that. The one thing that's not on here that I feel like should be is, so we've got convergent evolution, and then the other word that you sometimes hear is divergent. And divergent is like that adaptive radiation that we talked about. So the idea that you start with one species, but then, some of us are good runners, some of us are good swimmers, some of us are neither. But for those ones that are better swimmers, we're probably going to go, towards areas that are more aquatic, and those that are better suited for running, I don't know, would go to more areas where they could run. But the point is, you start off with one species, and then you kind of diverge out, or you radiate outwards. But that shows a common ancestor as opposed to convergent evolution, which does not. All right, let's get rid of that. I talked about that. All right, we can also look at molecular homologies. So, and there's lots of programs to do this now that help to look and say, okay, well this molecule is very similar to, it might be a different molecule, but it seems to perform, or it seems to, structurally, it seems to be like the molecule in another organism, but it might have a different feature in terms of what it actually does. So again, the structure is the same, but the function is different. It goes back to that one bone, two mode metibone. So we could have same or very similar DNA sequences, but now they're coding for proteins that do different things. They indicate a similar background or similar ancestor, but now they're doing something different with that particular DNA or chemical. Okay, so we can use these shared characteristics, we can use shared characteristics that are identified to help us construct these phylogenetic trees. So let's talk about the difference between what is called a shared ancestral character, which developed in the group, the ancestor, and then derived. And the derived is unique in that it's not shared by everybody in the ancestral group, but instead it's only shared by a group, a subset that has evolved to have a unique trait that or character that was no longer there. That is there that what was not there in the original ancestor. I'm going to give you an example of that will hopefully help. So let's look at one of these trees. So this is called a character table, and then we have a bunch of taxa. Taxa are just groups of organisms. If I say bass, there's lots of different bass fish. There's not just one type of bass. There's not one type of frog. So this is just groups or taxa that we're talking about. And we can represent these organisms having either a trait or not, like a binary system. So if you don't have a trait, it's a 0, and if you have a trait, it's a 1. So the lancelet is a very old species of fish. It's aquatic, and it doesn't have a backbone. It doesn't have a hinged jaw. It doesn't have limbs. It doesn't have an amniotic sac around its egg, and it has obviously no hair. So this one here, this is kind of the odd man out, and we actually call that the out group. And yeah, it actually does say out group right there. Because it shares none of the derived traits, and all of these over here, these characters, are the derived characters. It doesn't share any of those because that is the that's the low man, and that's this guy is going to come off first of all because it shares none of those traits. The next thing you're going to look at is the next group of organisms that has the fewest common features, and in this case that would be, they've nicely organized this so you don't have to think about it. The lamprey. So the lamprey has a backbone, but it doesn't have it does have a jaw, but it doesn't have a hinged jaw. It's really weird. It actually looks like a whole bunch of, like, that's the mouth and these are the teeth. That's kind of a bad drawing, but there's no hinge on it. They're just little guys here. That's a lamprey, and they don't have any hinge. They obviously don't have limbs. They don't have an amniotic sac, and they don't have hair. So then I put a little line here, and that is a derived character that is going to separate out the lampreys from the rest of the groups or the rest of the taxa. So the rest of those all have a backbone, okay? And then I can do the same thing for hinged jaws. Oh, sorry, that was the hinged jaw one, not the vertebral column. That was the lancelet, my bad. Let me just go over that again. So the vertebral column, the backbone, the lancelet doesn't have. So it's the first guy to come off because it doesn't have any of those characteristics. And then the next one is the lamprey comes off because it's got a jaw, but it's not hinged. Next one to come off is bass, don't have any limbs. Sorry, guys. And then it just keeps going. Okay, so we can kind of differentiate. And again, we don't know how long ago bass and frog separated. We know that they have a common ancestor at some point. Same way with turtles and leopards, and I would guess that turtles and leopards, obviously, it's been a while since they evolved from each other, probably longer than frogs from the bass. But I don't know. Point is, it's not showing us that relationship. So next slide is showing what we just went over. The outgroup is the one that has the fewest of these characters in common, and the in-group is everything else that's being studied. The outgroup is the group that has diverged before the in-group has, and that's really all we need to know there. Okay. Now you go on here, and I talked about this before, but I just want to bring it up again. Phylogenetic trees are hypotheses. They are not, they're best guesses based on the data that we have, but they're not telling us, they're making some predictions about what an ancestor might be, but they're not etched in stone for any way, shape, or form. And depending on what character we use, we can come up with very different relationships. So. But then we can use those to study and try and figure out how things might be related or not related. All right, here's another one. I've got some lizards and crocs and all these guys and how these are related to each other. I don't think we need to. Okay, so we can look at birds, and we can see that they are actually fairly closely related to dinosaurs. And that is taken from the fact that birds and crocs both have four-chambered hearts. They both sing. And believe it or not, crocodiles do nest. They build nests, and they brood, meaning they watch over their eggs before they hatch. So these are all suggest that these two are more closely related because this group of dinosaurs also had or seem to have those characteristics as well. So there's that. Okay, so far we've been talking about structures, homologous structures, and similarities and morphologies. But we can also look at genetic information. We can look at an organism's genome. We can compare their nucleic acids. We can compare their amino acids, their proteins, and start to trace back an evolutionary history there too, as well. In particular, we can look at how many times and how similar genes are, so we can look at the similarities in gene sequence, and we can see, the more similarities you are in gene sequence, then the more homologous and more closely related those particular groups or those particular organisms must be. Okay, so if we look at homologous genes, we can also look at duplicated genes. Another fun fact about duplicated genes, genes do copy themselves over time, so you don't usually have one copy of a gene. You usually have, for whatever trait it is, you'll have many of them. And the older the gene, the more copies you will have of it. And that also leads, we'll see down the road, to the idea that you can have those genes then can become mutated. So even though you've got functioning copies of a gene to produce a protein, there could be a mutated copy that could become a protein for something completely different. And we have seen that where some of those genes get mutated, and they still work, but they work in a different way that no one ever thought they would work like. So gene number and complexity of an organism is not strongly related. Turns out that humans only have about 4 times as many genes as yeast do. But a lot of that has to do with the ability, I remember we talked about exons and introns and how exons are able to be changed depending on what cell type you're talking about, and so that is one reason that the size of the genome is not related to how complex an organism is. Okay, let's see here. I wanted to see what else we need to talk about. Early taxonomists, this is in section 26.5. Early taxonomists classified all species as either plants or animal. Later we came up with 5 kingdoms. And then more recently, because of support from this whole genome sequencing and looking at all the DNA, looking at all the genes, all the sequences of A, C's, T's, and G's, we realized that we really should have, on top of kingdoms, we should have these domains. And we talked about these, the bacteria, archaea, and eukarya. The interesting thing about these is you think that you've got bacteria and you've got archaea. They're both prokaryotes, right? So they must be more closely related to each other than they are to eukarya. And it actually turns out that when we looked at some of that information, when we started looking at their genomes, it turned out that archaea and eukarya, are actually more closely related when you look at their genes than archaea is to bacteria, saying that the 2 prokaryotic domains are not as closely linked. They don't share as close a common ancestor as eukaryotes do with archaea, the domain archaea. And that's just kind of cool and weird too. And it also emphasizes what is called horizontal gene transfer. And this is the movement of genes between organisms that are in different domains. And this can happen through things like plasmids. Plasmids could have been taken up, or you can have infection, you can have endosymbiosis. And so there are ways to get genes into our genome and bacteria to get them into their genome that causes us to have more similar genes than you would ever think. You wouldn't think that we were, closely related to the prokaryotic domain more closely than bacteria is to archaea. Okay, I'm going to stop there, and I am going to assign you a worksheet that shouldn't take you very long to do. That is on cladograms, or these phylogenetic trees. And the reason I'm doing this, if I can find there it is. The reason I'm doing this is because cladograms, phylogenetic trees, are going to be important on the test next Friday, and they are definitely something that College Board in years past has liked to test you on. So I'm going to ask you to first analyze a cladogram, again, phylogenetic tree, and then I'm going to ask you to create your own cladogram after that. And then we will go over that on Monday if you have any questions about it, because you should be prepared to be able to interpret cladograms as well as perhaps make your own. Okay, so I want to make sure that you understand how to do that. So that's it, and I hope you all have a great weekend, and I will talk to you on Monday.